discussed a little bit about the difference between bacteria and archaea. Does anybody remember what the difference between those two things is? Main difference between bacteria and archaea?
How about a defined nucleus?
No, they both have neither of them really have defined nucleus. Prokaryotes don't have membrane-bound organelles like eukaryotic cells do, so that's not it, but that's good. Anybody else have any ideas? Mitch?
Bacteria have a flagella to move.
Some of them do, yep, that's true. Anybody else? Okay, well, bacteria cell walls contain peptidoglycan, which is going to be a big topic for today. That's kind of going to be the basis for the activity we do later in class. And archaea they don't have peptidoglycan. So if you try to stain them, you won't be able to stain archaea like you can bacteria. And another difference is that there are there's different lipids in the plasma membranes, and then the ribosomes are different. Ribosomes in archaea look more like the ribosomes in eukaryotic cells as opposed to prokaryotic cells. So we're going to talk about the structure of prokaryotes today. You can see some of the words on the slide are in blue. Those are the vocabulary words for this part of the chapter, so make sure you take note of those when we go through those. All of them are important, obviously, but just take a few more seconds to make sure you understand what those mean. Does somebody want to tell me what a chromosome is? Rebecca?
It's all the DNA that's bound around the. It's just very, very tightly wound DNA.
Yeah, exactly. So the chromosomes in bacteria are structured a little differently than the chromosomes you'd see in like our individual cells. In this picture, the chromosomes are in this blue stuff in here. This is called the nucleoid, which is the area in the cell where the chromosome is. So this blue stuff is the chromosome, and like this area where it is the nucleoid. This is one big circular chromosome right here. It's not individual ones like we have in our cells. We have 46 different ones. This one is just one big one. So, and then as you can see, there's this yellow part around the cell. This is the capsule. It's a polysaccharide layer that's secreted by the cell, and it helps to protect it from drying out, and it also protects, just protects the cell in general. And it also keeps antibiotics from getting into the cell, because some antibiotics will attack the cell wall, which is the green part on this picture, and the capsule keeps the antibiotics out, and it also keeps white blood cells from attacking the bacteria. When you have an infection, your white blood cells attack the bacteria, so the capsule helps to keep them out of there. And then the pili are these hair-like structures around the cell, and these help the bacteria attach to different surfaces, and they also help bacteria transfer genetic material from one bacteria to another. So there's two different types of bacterial reproduction, which we'll talk about in a later lecture, but two bacteria can attach by these pili to each other and then send their genetic information from one bacteria to the other. And then the size, bacteria are really tiny, really small, microscopic organisms. The nutrients, because they're so small, can diffuse through the cell really easily, which is good for them because they don't really have, there's different ways that they metabolize things, but being really small, it's easy for the nutrients to diffuse throughout the cell. So characteristics of bacteria, we're going to talk about two of them today, the shape and then the cell wall of the bacteria. So there's three different shapes that bacteria can be in, or are usually in. There's the cocci, bacilli, and spirilla. As you can see, cocci are circular, bacilli are like rod shapes, and then the spirilla or spirochetes are spiral-shaped bacteria. Those we don't see very often. When you look under microscopes at bacteria, you're going to see the cocci and bacilli a lot more often than the spiral ones. Nowadays, most bacteria is identified through molecular techniques, but before all that technology came about, the way they identified bacteria was by the shape and then their cell wall. So the cell wall of bacteria contains peptidoglycan, and depending on the type of bacteria, it contains different amounts of peptidoglycan. So this pink part right here is the peptidoglycan on this one, and then right in here is the same peptidoglycan. This, the gram-positive means that these bacteria, if you stain them with a gram stain, will stain purple, and the gram-negative means that they will stain pink. And this is because of the amount of peptidoglycan and because of this lipid layer that you see. When the bacteria is stained, the stain that you use is kept in the cell, in the peptidoglycan, and then it changes the color. And this lipid layer in the gram-negative side doesn't allow the original stain to seep in and get in here as well, so they stain a lighter color than the gram-positive ones. So, and I have some pictures here. These are gram-positive organisms. You can see that they're purple, and they're also cocci. They're little circular organisms, and they come in groups, as you can see. They come in pairs, and they can also come in single, just one, like here's one right here. And then gram-positive cocci like this could be, how many of you have ever had strep throat? The bacteria that causes that could look like this. There's also a lot of other organisms that are gram-positive cocci. And then these are gram-negative bacteria. They're bacilli, rod-shaped bacteria. And this could be, there's a lot of rod-shaped bacteria. In the last couple of years, we've heard a lot about E. coli outbreaks and food poisoning. E. coli could be one organism that looks like this. And then this is just a comparison, so you can kind of see there's some gram-negative and some gram-positive bacilli in this picture here. So does anybody have any questions, especially about the gram-positive, gram-negative things that we just covered?
I had a question about why antibiotics work if you said the cell wall keeps them out.
How antibiotics work?
Or
Why they work. Some antibiotics attack the cell wall of the bacteria, but then there's other types of antibiotics that attack other parts of the bacteria. They don't go for the cell wall. So there's different ways that bacteria, or that the antibiotics get into the cell. And not all bacteria have a capsule around the cell wall, only some of them do. Anybody else have any questions? Then we're going to go into the lab, and we're going to look at some bacteria.
This is microscope 1. What do you see?
Absolutely nothing to see that,
Oh, so we're going to rotate microscopes. We're just going to start at 1.
It takes a second for the light to turn on the microscope, so, and if you can't find anything, let me know and I can try and help you. Some of them are better focused than others. Right. If you want, you can. Yep, go ahead.
Gram-positive
Yeah, I focused it a little bit, but
Yeah, just like one.
you take a look.
Yep, there's a pink background. Anything else? If it wasn't moved, there's also a white blood cell in there. It's kind of hard to see, it's kind of in the pink background, but white blood cells do stain pink. And how about this one? Anybody who did microscope 2, what did you guys see here?
couldn't really tell.
Gram-negative though.
Gram-negative,
Good, yep.
they're kind of roundish.
Yeah, the background is really pink. This one is actually the one that I kind of had a hard time finding what I was looking for, so it is, the background is definitely pink. And then how about microscope 5? You guys see back there?
Pink hot dog rice.
Anything else? Did anybody see anything
like
different?
Purple blobs or streams.
Yep, they're
Oh, there's little like round ones too.
like big? Or you mean like little bacteria?
There's little bacteria.
Yeah. Okay. Good. and you guys are free to go.
Turn on the microscopes.
Yeah.